We'll move to our next speaker now, who is Dr. Victor Hernandez-Rocamora, also from Newcastle University. I don't know if there's potentially a link there, and he studies proteins involving peptidoglycan synthesis in E. coli, which is involved in what he's going to be talking about today.
Hello, everyone. Thanks for the nice introduction, and thanks for giving me the opportunity to present my work. So today I'd like to talk to you about a new real-time assay that we've developed to monitor the activity of PVPs in a membrane environment. I'm working in the laboratory of Valdemar Vollmer, so no connection with the previous, with Michael Hope. But anyway, so as you probably know, peptidoglycan is a very important layer of bacterial cell walls that it's important to keep the cell shape and to protect against turgor. And it's composed of glycan strands that are connected by short peptides. This polymer is synthesized on the outer side of the cell membrane from precursor lipid 2 that it's polymerized into steps. First, the glycosyl transferase reactions produce glycan strands that are then interlinked in transpeptidase reactions. And these two steps are inhibited by antibiotics that mimic lipid 2 parts, like minomycin that mimics the sugar and lipid part, and beta-lactams that inhibit the transpeptidase by mimicking the diala at the end of the peptides. So the molecules that we're interested, the proteins that we're interested in inhibiting are class A PVPs, and these are proteins that can catalyze both glycosyl transferase and transpeptidase reactions. And we are interested in gram-negative enzymes. So in these organisms, class A PVPs are anchored to the cell membrane by a transmembrane helix, and they contain a regulatory domain in between the two catalytic domains. That it's important because in gram-negatives, these proteins require the interaction with the regulators that are anchored to the outer membrane, which are lipoproteins, and interact with this domain. And then without this interaction, there's no activity in the cell. So in this presentation, I'll be introducing first results with PVP1B from E. coli and its activator LPOB. So we want a new assay that it's real time, that can report on both glycosyl transferase and transpeptidase activities, hopefully using the natural substrate lipid 2, and that can be adapted to lipid environments and taking into account also the regulation by activators. So we use FRED to develop this assay, and for that we need two probes, that a donor and an acceptor. So the emission of the acceptor has to, the emission of the donor has to overlap with the absorption of the acceptor. And if there's the two probes are sufficiently close, there will be emission of the acceptor when you excite the donor. So we attach these two probes to the third amino acid in lipid 2 to this position, and so we created two molecules labeled with the donor or the acceptor. And because of the way that we are attaching the probes, that means that we cannot have transpeptidase activity because we're blocking one of the residues involved in these reactions. So we need to add a third component to our reactions and label lipid 2 to have transpeptidation. So and another reason to add unlabeled lipid 2 is because when you have only labeled lipid 2, we saw that the proteins cannot polymerize these compounds. We think this is due to the large size of the probes that prevent two labeled lipid 2 molecules to bind to the glycosyl transferase domain. So only when you have unlabeled lipid 2 present, then there's peptidoglycan synthesized. So we prepared reactions with all these components, the enzyme and the three types of lipid 2. And then we observed that indeed FRED appeared. So we saw emission of the acceptor when we excited the donor. And this FRED signal was inhibited if we added minomycin, which blocks glycosyltransferase reactions. So no peptidoglycan is synthesized. In fact, we could monitor these activities in real time by calculating a ratio between the thread signal and the donor signal. With this methodology, we could detect the activation of this activity by LPOV, the activator, and the accelerated reactions, and also the effect of inhibitors like ampicillin that blocks transpeptidation. With ampicillin, what we saw is that the signal was affected enormously, though the kinetics was the same. And why is product that inhibits transpeptidation affecting so much the signal? So we think this is due to the fact that because no two labeled molecules can be inserted adjacently along the same strand, then the distance has a minimum, the distance of the probes, and this produces a low thread, and only when there's cross-linking, the probes can get close together to produce high thread. So all these results were done in detergents, but what about the membrane? So we took advantage of the fact that these enzymes cannot polymerize the labeled compounds on their own, so we reconstituted both the labeled compounds and the enzyme in the same liposome, then started reactions by adding unlabeled lipid 2. We performed assays in this way, and again, we could monitor the activity of the enzymes in real time, and we saw inhibition by glycosyltransferase inhibitors, activation by LPOB. Both probes were incorporated into PG, and we also could detect inhibition with ampicillin. And one important thing is that reactions in liposomes were slower than in detergents, and the activity was to have a decent activity, you require the activator. We think this reflects what happens in the cell. So it's important to test your reactions in a more physiologically relevant environment. And not only we did this assay with E. coli PP1B, but we also used from PPs from other gram-negatives like Acinetobacter baumannii and Pseudomonas aeruginosa, and in both cases we could detect their activity in the presence or absence of the activators. So in summary, we developed a new real-time assay to monitor glycosyltransferase and transpeptidase reactions of class A PPPs. This is the first time that such assay has been developed, allowing proteins, reconstituted proteins, to be monitored, and hopefully we were hoping to use this to screen inhibitors in a more physiological environment. So I'd like to thank my co-workers and our collaborators in Austria and the Netherlands, and thank you for your attention. And if you're more interested, we've published, we've recently, we submitted this to a journal, but we have these also available in this preprint server. Thank you very much.